Today's lesson is Investigating Diversity. That's section 10.4 in your textbook. So could you please make sure that you are reading through that section, making summary notes on that section as part of this week's task. Please always look at the learning objectives at the beginning of the chapter because that really helps you focus on what you need to know. Okay, so we'll start off with a starter. So the starter activity is for you to calculate an index of diversity. So this is the table that is present on your starter activity. The actual question itself is as follows. So it's the right one. Yeah, it's this one here. So if we sort that out. Okay, so you will have a document, either a PDF or a Word document. So here is your data. Table shows data collected in the Word. So here it all is. First, there's a question about what's the importance of sites being chosen at random. Then you're given your index of diversity formula, and you have to calculate the index of diversity for the n-sex court in the Wood. And then there are a number of other questions. This should take you approximately 10 minutes to complete. So give that a go and then come back to the PowerPoint. Okay, so sharing is paused. Let's close that. Okay, here we go. All right, so 10 minutes for that, please. Okay, so you've completed that. The mark scheme will be attached to the actual document. So please, can you mark your work? Any questions for that, please let me know on the chat. If you're struggling to calculate the index of diversity, if you're not getting the number that they get, please let me know. Ask on the chat. I can make a video to show you how to do it. All right, so we saw in a previous lesson that there is a relationship between different living organisms, and that relationship can be shown in the form of something called a phylogenetic tree. So the phylogenetic tree, the picture that you're seeing here, is in fact a doodle, or drawing, should I say, from the Origin of Species, drawn by Darwin when he was off doing his research. And so he came up with this idea of the relatedness of all things. So the fact that you might have a starting organism, and so number one over here, and that would live for a certain period of time, and then something would happen. There'd be a mutation in the DNA, and circumstances would change, environment change, and you have a speciation event. So you have these branches going off, and then that might happen again over time, and might happen again over time. And so it goes to demonstrate that there is a relationship or a connectedness between different living organisms. And when he did this, he based all of his phylogenetic trees on the similarities and differences in their physical appearances, because he couldn't do anything else. He didn't have any genetic information. He could only look at the animals. So this is the kind of thing he did. He looked at animals and he said, right, here we have a bunch of animals. What do they have in common? Well, they've all got a backbone. They're all chordata. They all have this spinal column. What else do they have in common? Well, they all appear to have 4 limbs. They're tetrapods. Okay, so fish, frogs, rabbits, lizards, birds. So seeing homology between all of these animals, all of these vertebrates, clearly they're not the same. That one's got feathers, that one's got scales, that's got fur, that's got a slimy skin. They are all very, very different in other respects, but they do have similarities. What evolutionary biologists would have done is they would have looked at the things they have in common. So they might look at the forefoot or the forearm of the animal and find similarities. And they can do that they can find similarities in the leg of the frog, in the forefin of a lobed fish, in the leg of a rabbit, in the leg of a lizard, and in the wing of a bird. So we call these homologous structures. Remember the prefix homo means same. They're homologous, they are same or similar. In the same way when we look at plants, this is a stereotypical drawing of a leaf, but there are so many different adaptations of leaves, different types of leaves. So this is a leaf as we might draw it if we were going to draw one, but these are all adaptations. These are all homologous sort of structures basically, but they have been modified to have different functions. Okay, if you are going to build a phylogenetic tree based purely on the observable homologies, this is the kind of thing that you might come up with. So you might start with, okay, you might think, right, this is our first vertebrate. So our first vertebrates at some point in evolutionary history, so if we're following up, this is a timeline, call it, in some point in evolutionary history, there would have been a mutation and there would have been a speciation event. And this branch would have gone off to make the cartilaginous fish, like sharks. So they're slightly more ancient fish types. Sharks and rays are cartilaginous. And then we have the development of the bony skeleton on this arm. And again, there might have been another speciation event, so mutations happening here, change in environment, speciation event. This branch goes off to make the ray-finned fish. So these are our bony fish. So this is like our sort of typical cod, hake, whatever, goldfish, you know, the non-ray, non-shark fish. And the other would have gone off to be the branch from which other four-limbed animals arose. Again, evolutionary time, mutations, changing environment, speciation events happening. And so we'd have the amphibians, and then maybe here's the amniotic egg branch. And so we go. Okay, so you can follow that and you can see the logic to that. But as you know, we now have masses and masses of genetic information. We have masses and masses of DNA for lots of living organisms. So we have a human genome, we have genomes for lots of primates, we have genomes for loads of different living organisms, plants and animals. And one way that we now figure out the phylogenetic relationship between living organisms is by taking their DNA sequences and comparing them. We don't take the entire DNA sequence, we take a DNA sequence of a gene that we are interested in. So there is a protein that is involved in respiration. It's found in the cristae of the mitochondria called cytochrome C. Well, that is used by the majority of living organisms because the majority of living organisms respire in a similar way. So if we take the DNA sequence of cytochrome C and we compare it from lots of different living organisms, we can see how similar they are to one another. So using the similarity, we can then build a phylogenetic tree. There are no real examples in here, but this is just showing you. So here we can see the T is conserved, the G is conserved, the A is conserved. Here, for whatever reason, in these sequences are now missing. So there's been a deletion here. Okay, and so you can see that this computer program, because this will be done by a computer programmer, by a computer program, has created a separate branch for these living organisms because they are more similar to each other and more dissimilar to all of them. Okay, and another, if you look at this, you'll see there's other dissimilarities, areas of dissimilarity, and the computer program will use that to work out how related the living organisms are to one another. Okay, so that's using genetic material, using the DNA. What's actually even more useful is when we compare the sequences of the proteins that come from the genes. Okay, so here is a protein that is being compared for a variety of living organisms. So what's interesting about proteins compared to DNA is we know that the DNA code is degenerate, so we know that some mutations in DNA will give the same amino acid. So there will be no effect to the actual protein sequence. But what's interesting in this is also the type of amino acid, because you get amino acids with different chemistries. You get polar and non-polar amino acids, and it's interesting to see how they might change for different living organisms. So if we have a look at these particular sequences over here, we can see that there's a lot of similarities. So R, F, R, Y, K, they are single letter representations of a single amino acid, okay? And so we can see there's a lot of similarity. There's a human, there's a, not quite sure what that is actually, it looks like a armadillo or something, and then we've got chickens, and we've got frogs, and we've got various fish. You can see how well amino acid sequences are conserved through living organisms. You know, we do start to see some differences, but a lot of conservation. So we can see there is a relatedness between all living organisms. The more similar, the more similar the sequence, the more related the living organisms are. And we can see here we're going down to slightly more simple living organisms, and we can see they actually don't have this domain of this particular protein. Okay, nematodes don't appear to have the protein at all, and so we can see that, you know, that in terms of relatedness, we can come to different conclusions. So that when we get down here and we compare the porifera to the higher order sequences, that they are less related because they are missing quite a bit of the sequence. Not quite sure how this fits into the homology, but it could be based also on other bits of the protein sequence. We're not saying. Either way, hopefully you get the idea. The more similar they are, the more related they are in evolutionary history, so the closer they will be in the phylogenetic tree. Okay, so this gives us another sort of representation of exactly the same idea. So here we have a y-axis that says number of amino acids that differ from the human hemoglobin polypeptide. So this is comparing hemoglobin sequences of different living organisms. So if we compare the human with the rhesus monkey, we can see that there is a small percentage difference of amino acid, of the number of amino acids between the rhesus monkey and the human. So our hemoglobin is relatively similar to that of the rhesus monkey. Less similar, the mouse is less similar to human. Chicken is even less similar to human. Frog is even less similar to human. So we can see this relatedness decreasing. Lamprey is a type of a primitive jawless fish. And we are, in terms of the hemoglobin comparison, we are not, we do not share a recent common ancestor with a lamprey, because our hemoglobin sequences are significantly different. Okay, there are 125 sequences different, it appears. Before we end, I just need to mention this. This came up in your textbook. This is how we use immunological studies to study the relatedness between living organisms. This is quite an old-fashioned technique, and it wouldn't be used very much now, but you need to understand it because it is in your curriculum. So what one might do here is take some human serum. So take some blood, remember centrifuging, you spin it so the cells will pellet to the bottom of the tube, and then you just take the plasma off the top and you leave the cells behind. Take that serum, inject it into a rabbit, poor rabbit, and the rabbit will make some antibodies to that serum. Then the scientists will extract the blood from the rabbit, spin the rabbit's blood, separate out the cells, and then take the plasma serum from the rabbit and mix it with the blood of other species. Now, the rabbit serum, the rabbit has made human antibodies in its blood. So the rabbit has got human antibodies in its blood. If we add human antibodies to human blood, those antibodies will stick to the antigens in the human blood and they will precipitate out. Okay, they will stick because they are complementary to human antigens because they are human antibodies. If we have to mix these human antibodies with blood from a gorilla, we will get some precipitation, probably quite a bit, but not as much as with the human because gorillas are relatively similar to us. So we might share some antigens. So the antigens that we share will get precipitated by the human antibodies. And then we could do the same for baboon blood. And the baboon blood might also precipitate somewhat, but less than the gorilla, which is less than the human. And so we might go, a lemur, which is a type of a primate, but not related to us, might show a small amount of precipitation. Rat might show a really tiny amount of precipitation because it's unlikely we share very many antigens with a rat. Okay, so it's an old-fashioned technique, but you should be aware of it. Okay, so before I end off here, this is a phylogenetic tree of fish. Now, some of you might listen to a podcast that is called There's No Such Thing as a Fish. And that comes from the fact that people who study evolution believe that there is no such thing as a fish, because when they look at the genetics of different groups of living organisms, they can put them into phylogenetic trees, and the phylogenetic trees make groupings called clades. And a clade is hang on a minute, I've lost my let's see, a clade is a grouping. So say for example this, what looks like a sort of a candlestick, that there would be considered a clade. So it's a clade is a group of animals that share a common ancestor. Okay, now groups of animals usually all belong to the same clade. So for example, all mammals belong to the same clade, all reptiles belong to the same clade, all amphibians belong to the same clade. But not so with fish. So if we go back in time to the deep dark evolutionary past, and we have our first speciation event, we get hagfish, the group of hagfish coming off here, and then speciation here. We have lampreys coming off here, and then we have another speciation event. We have our sharks and rays. Remember we said those cartilaginous fish are quite old. And then we have another speciation event. And up here we have the lobed fin fishes. But in this group as well, we have tetrapods. So all human life, all bird life, all reptilian, all amphibian, all of the other vertebrate groups with 4 legs are in this group over here. So the reason that people say there's no such thing as a fish is because there is no single clade for all the fish. We are essentially in the fish clade. Okay, so so that's where that idea comes from. I just also thought I would share with you some links on hagfish and lampreys, because hagfish and lampreys are jawless fish, and they are ancient types of fish, and quite unattractive really. But this is in conflict with this idea. So you could see that if we were just using physical homology, the fish sit in their own clade. That would be a fish clade, that would be a fish clade. But you can see using genetics, we are basically in the fish clade. Okay. Oh yes, and that is I thought I'd show you this. This is a lamprey mouse. So a lamprey is jawless. So it's got this sort of sucker mouse with these sharp bits on it. So have a look at that if you're interested in different types of fish. And then very final thought on the evolution of viruses. I just thought maybe in context of coronavirus and how viruses continue to perpetuate themselves. If we think about viruses, if they're very, very virulent, so very, very, very dangerous, people get very sick and do not go out into public, and they will then not spread the disease around. So something like Ebola would would have a very high mortality rate, but it would probably, with good isolation, would be easily contained. Whereas a less virulent virus, so something that's really, sort of like the cold or the flu, that's not going to kill you, but it's going to make you feel quite unwell. It's going to have you walking around, coughing and sneezing. This is going to have a better sort of evolutionary chance, as it were, because there's a good chance for transmission. So it's interesting to think about coronavirus, and with coronavirus, it's in this context. Okay, all right, so I think that is it for today. Apart from this, could you please, now that you have done all of your work on looking at investigating the relatedness of living organisms, can you do this question, and then can you mark it afterwards? The mark scheme is attached. Okay, all right, lovely. Thank you very much.